So good afternoon to everyone. So first, thanks to the organizers for putting together this fantastic mix of talks and topics, and also for giving me a chance of being part of it. So what I'd like to talk to you about today is the way in which breathing can influence freezing behavior in the mouse. So of course, all of us have experienced that when we're in an emotional state, our bodily state is going to change. For example, right now, I've just realized quite how big the room is, and my heart rate has gone up just a little bit. And of course, given the topic of the symposium, I'm not going to really have to argue very hard for the idea that information also flows the other way. That is, bodily states can influence emotional states and emotional behavior. And throughout the past 3 days in flies, humans, mice, we've seen many examples of this, particularly in Sarah's talk, as she just pointed out. And I briefly just wanted to highlight that, one of the ways in which these ties between the body and emotional state becomes manifest is in pathology. Actually, as Sarah just mentioned, bodily hypersensitivity seems to predispose to anxiety, but also at the therapeutic level, there is vagus nerve stimulation that is a potential therapy for depression. However, in spite of these ties, in humans, it's been very hard to really find a smoking gun that's linked lack of bodily feedback to a lack of emotional response and arousal. There are some cases where, for example, in sympathectomy, people have experienced loss of emotion. But if we take this study, for example, in spinal cord lesion patients, so where, of course, there's a massive loss in feedback from the body, patients actually report quite ambiguous emotional states. So if we just take the first patient here, what he or she says is that, I believe I feel a little less tension when I get mad. I don't shake so much. It's easier to control. Maybe I get over it a little quicker. So it seems that there's some change. And then you see the patient sort of backpedals and says, well, maybe I didn't get that. Maybe I always got over it fairly quickly. And this is quite a general pattern, is that despite huge changes in bodily feedback, these people definitely still have rich emotional lives. What this underscores is that there's still, I think, an open debate about the nature of the causal relations between bodily states and emotional states. And I think a way of trying to understand the debate is to go back to the historical debate between James Cannon, because they sort of span the field of possible visions of what's going on. So to caricature them just a little bit, to simplify, we have the Jamesian's point of view, in which unique bodily states generate and in fact are emotional states, whereas all the way on the opposing view, Cannon questioned whether bodily states were specific enough to contribute to emotions and whether they actually played any causal role at all. And so I think it's helpful to try and understand these causal roles to separate two different problems out. The first is the problem of mechanisms. And basically what that means is, what is going on behind these arrows that I've drawn here? What aspects of emotions are being impacted, and what is the physiological mechanism? And the other aspect is specificity. That is, are there really specific bodily states that correspond to specific emotions and therefore can specifically regulate them? And what I'm going to show you today in the mouse is two studies that begin to shed a bit of light on these questions in the case of breathing. So we want to study emotions in the mouse, and unfortunately for rodents, the go-to behavior is fear. So the way we evoke this in this case is with classical tone fear conditioning. We couple a mild foot shock with a tone. Then when you present the tone alone, mice tend to freeze. That is, they become completely immobile. And what many people have now noticed is that whereas when a mouse is exploring the environment, it tends to breathe quite irregularly at around 10 hertz, when it begins to freeze, you get this lovely regular 4 hertz breathing that appears, as you can see here, by the very peaked histogram of breathing rates. And this is very interesting because we know in the mouse that breathing can really feed back to the brain and have an influence. So the way this works is that as the mouse breathes in and out here in gray, air is going in and out of its nostrils, and in its olfactory bulb there are pressure sensors that are going to respond to the air flowing in and out, and therefore entrain the olfactory bulb local field potential, as you can see here, really tracking breath by breath what's going on. And when the mouse begins to freeze, we get this very regular breathing, and therefore this very regular oscillation in the olfactory bulb, as you can see, by a very peaked spectra in the activity. And we know that the olfactory bulb can then feed this back to many regions in the brain. There's been a host of studies going back to the 90s and even more in the past 5 or 10 years, showing that it's present in many cortical areas. And so what I'm going to show you today focuses on the prefrontal cortex that we know to be an important hub for fear control in the mouse, and also since when we performed this study, there was already some data suggesting the 4 Hz oscillation could be found there. So if we record both in the olfactory bulb and in the prefrontal cortex while mice are freezing, what do we see? In the olfactory bulb, we see essentially what I've shown you before. As the mice begin to freeze, we get this very regular 4 Hz oscillation appearing. If we look now in the prefrontal cortex, we get essentially the same oscillation. And if we calculate the coherence, that is essentially, are they going up and down together, we see that in fact they are very coherent. And we know that this prefrontal cortex oscillation is really coming from the olfactory bulb, because if we remove the olfactory bulb, as you see here, the oscillation goes away. So this is all based on local field potential, which, although it's called local, is in fact often a rather ambiguous indicator of what's actually going on in a brain area. So we also recorded individual neurons in the prefrontal cortex to see if they were responding to this respiratory rhythm. So the way we looked at this is to take all the spikes of an individual neuron and see whether these spikes tended to occur at a specific phase of the respiratory rhythm. Here you can see an example where it's very strongly the case during freezing. You see the spikes tend to occur at this phase of the breathing, and it's a little bit less clear in the active state of the animal. And if we look at overall all the units we record in the prefrontal cortex, we find that around 45% of them, so almost half, are in fact entrained by this respiratory rhythm, and this is stronger in the freezing state than in the active state. And once again, we know that the olfactory bulb is necessary for this entrainment, because if we degrade the olfactory bulb, here using a pharmacological treatment, we see a drastic reduction in the number of modulated units. And those units that we find that are still modulated have this rather unconvincing modulation. So what have I showed you so far? When a mouse begins to freeze, it changes its breathing to a very regular 4 Hz that feeds back via the olfactory bulb to the prefrontal cortex, where this oscillation is going to modulate neural firing. But this, of course, raises the question of whether this sequence of events is actually going to impact the mouse's behavior in the fear task. So first, let's look at the bulbectomy animals I presented. So in these animals, if we just look at the amount of freezing they display, we see that compared to their sham controls, there's a drop in freezing levels basically throughout the test session. However, bulbectomy is, of course, quite a massive intervention. As you can see, there's a big bit of brain missing here at the front. And so we wanted to shift to something more precise, and particularly more precise in time, where we could really aim our manipulation at the time where the mice should be freezing. So we shifted to an optogenetics approach. We expressed in the interneurons of the olfactory bulb channel rhodopsin, which allowed us to impose an extra oscillation on top of the 4 Hz oscillation. As you see here, before we stimulate, we have this nice 4 Hz oscillation. We begin to stimulate at 13 Hz, and essentially what we're doing is we're scrambling this temporal organization in the olfactory bulb. So we're cutting off the regular respiratory rhythm from getting to the rest of the brain. And when we do this, what we see in our channel rhodopsin mice is also a reduction in freezing levels. So what I think this shows is that clearly the feedback of the respiratory rhythm is causally involved in regulating fear behavior. However, this left us with a little bit of a conundrum, because on the one hand, it seemed as if freezing was necessary to generate the 4 Hz behavior, but our behavior results also seem to suggest that the 4 Hz itself was in turn necessary for freezing, and so this seemed a little bit circular. So we went back to our data and decided to analyze a bit more carefully what was going on. So we made a very, very simple model of behavior. It's a 2-state Markov model. Either the mouse is in a freezing state or in an active state, and it goes from one to the other according to a probability. So we have one parameter that corresponds to the probability that the mouse is going to initiate its freezing, and another parameter that corresponds to the probability that it's going to stay freezing once it's started. So what we call freezing maintenance. And we can just look at the values of these 2 parameters in our data. So the first thing we see is that if we take initiation, either for the bulbectomized mice or the optogenetics mice, we don't see any change in the initiation parameter. However, if we look at maintenance, we see a drop in both groups. So what this means is that the mice, without the respiratory feedback, are really struggling to maintain stable freezing states. And this allows us to answer our question of the sort of apparent circularity, because in fact we see that there is a brain-body-brain loop with 2 arms. The first arm that we believe corresponds to initiation, that it is not affected by blocking breathing, is likely mediated by the periaqueductal gray and the amygdala, because we know from the literature that if you stimulate these regions, you induce freezing and you induce changes in bodily state. So once this initiation has happened, the animal is freezing, it begins breathing at 4 Hertz, and this is going to recruit the maintenance arm of the loop, which, via the olfactory bulb in the prefrontal cortex, is going to basically allow the animal to decide how long it stays freezing. So this suggested to us that we should see in the prefrontal cortex data some indication that this brain region was more closely involved in freezing maintenance as opposed to initiation. So to do this, we went back to the neural data I mentioned earlier. So if you look in the prefrontal cortex, what you will see is, if we take all the units we recorded here, overwhelmingly they tend to be inhibited during freezing. A few of them pop up being activated. Now, if the prefrontal cortex is specifically involved in freezing maintenance, we expect it to have a strong relation to freezing offset, because this is the time when the animal is deciding, do I maintain or do I switch behavior? But not to have such a strong relation to freezing initiation, because this is not controlled by this arm of the loop. So what we decided to do is to compare their activity at the onset and the offset. And to do this, we cut up our neurons into 3 different groups. The first group is neurons that are modulated by the olfactory bulb oscillation, so those that are really impacted by the respiratory rhythm. The second group in gray is those that are not modulated. And the third and perhaps most important group are neurons we recorded in animals that were bulbectomized, so where there was no contribution at all from the respiratory rhythm. And what we see in these 3 groups is at the onset of freezing, basically they're all behaving in the same way. But if we now go and focus on the end of freezing, what we see is there's a very strong offset response here in the modulated group that's weaker in the non-modulated group, and that very importantly completely disappears in the bulbectomized group. So what we see at the level of neurons is that the fact that these neurons are being modulated and entrained by the respiratory rhythm seems to allow them to be recruited and perhaps to help regulate this phenomena of freezing offset and freezing maintenance. So to conclude on this part, what we've seen is that if we look at freezing, mice of course become immobile, but they also change their bodily state. They begin to breathe at a very regular 4 Hertz activity that can feed back to the brain, influence prefrontal cortex firing, and regulate freezing by controlling the probability of maintaining this state. And so if we go back and try and put this into the bigger picture I started off with in the introduction, in terms of mechanisms, what we see is we do have a mechanism by which the body can influence the brain. It's the rhythmic activity of breathing in training neural oscillations. And we do have indeed causal evidence of the body influencing the brain, but having a rather specific influence on freezing maintenance and not initiation. And so going back to the James-Cannon debate, we see that we do have a causal influence, but at the same time, this Jamesian idea of bodily states generating emotion, in this case at least, definitely does not seem to be the case. And I think this fits with what's been seen in the mouse in other studies. For example, that Nadine Gogola showed us that vagus nerve isn't directly controlling whether the animal is afraid or not, but it's playing this more subtle role in fear homeostasis. And similarly, a study looking at the influence of heartbeat has shown that animals have to be in an anxious state for the heartbeat to have an influence. And I think this gives a little bit of insight into the sort of complicated interpretations of what happens in humans when they are lacking bodily feedback. Because what we see is not that these causalizations are ambiguous, but they are in fact specific and restricted, because the body, as I said, is not generating these emotions, it's regulating specific aspects of these emotions. And for example, what we should expect, and what you actually see if you go through the literature, is that people still experience emotions, but their temporal structure has been changed by this lack of bodily feedback. However, what I've shown you so far does not at all speak to the specificity of body states corresponding to emotions, because during freezing, we expect the mouse is afraid, but it's also immobile. So of course, the breathing could simply be changing because they've changed their motor state. So what we would like is to remove this motor confound, and now I'm just going to briefly show you some results where we think we have identified a bodily state that can be directly linked to emotional states. So to do this, we changed paradigm because we wanted to look at a wider range of defensive states than simply freezing that can be evoked by tone fear conditioning. So in this paradigm, The mouse is exploring a U-shaped maze, and it receives shocks in this arm over here. And so after a while, the mouse tends to avoid the shock zone and spend most of its time in the safe part of the arena. Now, what's interesting in this task is we see a very organized behavior for the mouse. If we take the maze here going from the shock zone to the safe zone and sort of linearize it, so we have more or less a distance from the shock zone, we see that we have a clear organization of behavior with explosive escape and jumping over here, risk assessment in the middle, and grooming over here. And this, for us, was very reminiscent of what has been seen in the ethological literature, where it's been described that animals organize their behavior depending on predatory imminence. So whether the cat is right in my face and about to eat me, or whether I'm not even sure the cat is there at all. And interestingly, this has been related to panic and anxiety recognized in humans, using pharmacological tools in humans, applying them to animals, and therefore mapping this scale onto panic and anxiety. So this was encouraging, but we had a bit of a problem, which is that freezing was everywhere. As you can see here, it occurs basically in all four corners of the maze, both in the shock and the safe zone. And I'd really like to emphasize that really the mice are completely immobile. This is real freezing. They're not moving at all, as measured by head acceleration. And so this raises the question of whether freezing is violating this sort of very structured behavior we have for the other parameters, or whether what seems to be a homogeneous state is in fact heterogeneous, because here we're just looking at the behavior of the animal and not at its internal state. So what does the data tell us? If we take the olfactory bulb spectrogram, so this is frequency and time here, and the spectrogram of the olfactory bulb, remember, is actually a proxy for the breathing of the mouse. So as the mouse wanders around the environment here, the mouse is breathing at 10 hertz, goes into the shock zone, gets a shock, and starts to freeze. And we see this breathing at about 4 to 5 hertz here. And here is an example of the data. The mouse then gets another shock, decides it's time to run away, goes to the safe zone, and freezes again. And you see that immediately, the breathing changes to quite a different frequency. We here instead have breathing at around 2 to 3 hertz. And here is an example of this signal. So we see that in the shock and safe zone, in fact, the breathing is really distinguishing these two types of freezing. And this is true for all the mice that we recorded in this paradigm. And although I'm focusing on breathing, I'd just like to point out that we see similar things for heart rate that is different between the shock and safe zone, and also for peripheral vasoconstriction measured by tail temperature. So if we put all this together, what we have is two states with identical behavior, so no motor confounds, but very different bodily states related to breathing, but also the cardiovascular system. And this suggested to us that maybe these are panic and anxiety-like states, which would really allow us to pin down these bodily states to specific emotions. And so to test this, we took a leaf from the ethologist's book and decided to try and use panicogenic and anxiogenic stimuli to see if they would also generate these two different kinds of freezing. So the first thing we did is we put a mouse in a completely blank box, nothing scary, and stimulated its dorsal periaqueductal gray, which is known to be a panicogenic. And at whatever voltage we stimulated, the mice tended to freeze, and they always freezed with this 6 hertz breathing, very reminiscent of what we saw in the shock zone. Now, if we do the same thing, but if we administer an anxiogenic, so here it's quite a strong dose of nicotine, again, the mice freeze, but here instead we have this slower breathing, reminiscent of the safe side freezing. And I'd just like to point out, we also tested this with anxiolytics and panicolytics that basically paint the same picture. Things are a little bit more complicated, so I didn't put it in here. We can come back to this in Q&A if anyone is interested. So I think what this shows is that in this U-maze task, we see freezing in 2 spots with exactly the same overt behavior, but very different internal bodily states for the mouse. And this, in fact, can be linked to a panic-like and anxiety-like state, as we would tend to define them in humans, thanks to these electrical pharmacological manipulations. And just finally, to go back to the sort of bigger picture I was trying to paint, what this shows is that in the mouse, in this instance, we do in fact have quite a good specificity between the body state of the animal and a particular emotional state, in this case, panic and anxiety. And I think this raises a bit of a challenge to go back to the question of mechanisms, because the question it raises is whether these distinctions in the body actually make a difference in the brain. That is, are the brain circuits sensitive enough to perceive and respond to these differences of 2 to 3 hertz or 4 to 6 hertz in the breathing, and therefore recruit different mechanisms and regulate differentially this panic or anxiety behavior. And so with that, I would like to point out that all of this work has been done in the lab of Karim Benchinan, who is really a great mentor. And both projects I presented were really done in tandem. The first project with Julie Lefort, and the second project with Baptiste Mahot, who is sitting right there, and who presented a poster on this actually yesterday. And I'd also like to thank the other lab members, the collaborators, and you for your attention.
Thank you very much, Sophie.